So here we are with a animated video to understand the process of translation or simply the formation of protein from mRNA. The translation process actually includes the presence of mRNA along with two ribosomal subunits, the smaller subunit and the larger subunit, and tRNAs carrying designated amino acids. On the top, you can see this green labeled tRNAs along with the blue colored amino acids that are attached to it. They're ready, and here comes our mRNA. And the mRNA has a cap which is labeled as methylated guanosine, and a tail which is just a long A, as you can see on the right hand side in a yellowish rectangular box. It is called a polyadenine tail, and just near to the cap is present our start codon labeled in a white rectangular box with the triplet codon AUG, and on the right side near to the poly A tail is that red rectangular box that signifies our stop codon, and just in between the start codon and the stop codon is the blue region that you can see here. This is our structural gene that contains the triplet codons that actually will code for the designated amino acids. So let's go ahead and let's see what happens. So as you can see, it is labeled mRNA, and here comes our smaller subunit of the ribosome, particularly 40S, and comes the larger subunit here, and it contains three sites called the exit site, the peptidyl site, and the acceptor site, or very shortly we can call it the EPA site of the larger subunit of the ribosome. Now this larger subunit of the ribosome is actually 60S. The smaller subunit is 40S and the larger subunit is 60S. Okay, so here let's go ahead. As you can see, this green structure that comes into the slide here, it is the release factor or a special protein that will eventually come to use at the end of the translation process. Now if we go ahead, as you can see that the peptidyl site or the P site of the larger subunit sits right above the start codon or the AUG of the mRNA. Similarly, the smaller subunit also sits below the start codon of the mRNA. Now the P site is open to accept a tRNA that contains the complementary anticodon or the anti-triplet codon to the AUG or the start codon of the mRNA, and here comes this particular tRNA with that anticodon USC, which is complementary to AUG or the start codon of the mRNA, and as you can see, it is carrying a blue colored amino acid here labeled MET or the methionine. Okay, so this is the first amino acid carried by the first tRNA, which actually comes and sits at the P site of the larger subunit of the ribosome. At the same time, the A site of the ribosome, the larger unit of the ribosome, is also sitting right above the next triplet codon of the mRNA, which is UUA. It means it can also receive another tRNA containing the complementary anticodon or the anti-triplet codon carrying its designated amino acid. So let's see which comes here. So you can clearly see that it's the tRNA containing the anti-triplet codon AAU, which is complementary to the UUA triplet codon of the mRNA carrying a particular amino acid. The next step is the entire ribosomal unit, both the smaller and the larger subunit, will actually slide to the right hand side of the mRNA, and let's see. So it's going. The tRNA from the P site, the first tRNA, it has to slide over to the E site, and the second tRNA which was at the A site slides to the P site. However, the amino acid at the first tRNA, which was the methionine, leaves the first tRNA. It breaks from the first tRNA and forms the peptide bond to the second amino acid present on the second tRNA. Thus, a dipeptide chain is formed at the P site. Now, the acceptor unit of the larger subunit of ribosome is empty again, so it is ready to receive another tRNA carrying another designated amino acid, which will be complementary to the third triplet codon UCA of the mRNA. So here comes that tRNA, and as you can see, it is carrying an amino acid here. And similarly, the process will repeat itself. The ribosomal unit will slide to the right hand side. The tRNA at the E side, the empty tRNA will again escape or leave the site. So let's see in the animation, it's leaving, and the ribosomal units will now slide to the right hand side along with the dipeptide chain, now forming the peptide bond with the third amino acid, forming the tripeptide chain. And as always, the acceptor site of the larger subunit becomes empty again, and the process will continue, as you can see in this animation, until it goes and reaches the stop codon of the mRNA. So here comes that point when the acceptor site of the larger subunit actually slides over the stop codon of the mRNA. Now, the release factor that you can see on the right hand side, which is the only one left in the screen here, that green oval shaped structure, it is the only structure that can come and bond to the stop codon of the mRNA, as it contains complementary anticodons. But the fact is, it does not carry any amino acid with it, as it is not a tRNA. It is just a special protein called as a release protein or the release factor. So eventually, as you see, it will come and sit over here. The empty tRNA will eventually leave the E site, and the ribosomal unit will slide towards the right, so that the tRNA from the P site leaves the amino acid and becomes empty and slides over to the E site of the larger subunit of ribosome. Now the polypeptide chain, as you can see, it is coming in contact to the release factor, but the release factor do not have any amino acid with it, so there will be no peptide bond formation here, resulting in the termination of the polypeptide chain. The empty tRNA leaves the E site, and eventually here we can see that the polypeptide chain gets released. Now the release factor has no other work other than to leave the P site, and the ribosomal units also disassembles from the mRNA. The mRNA eventually disintegrates. So we are left with the polypeptide chain here. This polypeptide chain will coil itself to form a complex three-dimensional shaped structure, and this complex three-dimensional structure of this polypeptide chain will determine it as a functional protein. So with this, we have come to the end of the translational process.